Okay, so that means I'm going through bit by bit with you to make sure that every segment here, you know, you understand, and you can master the concept and then answer the question. All right. So today, what we are going to do is uh, replication. Okay, the DNA structure, replication, and then end replication. Probably in 2 hours, you can only do this together with answering some questions. Okay.
Mm,
Now, for you, what is your role is your role is to ask question and stop me if you don't understand, and then
okay.
ask your questions. You know, whatever that you don't know and stuck one when you study is to ask because this is your lesson. Okay, so for one-to-one means what now? One-to-one means that the time is actually yours. The pace is also yours. Okay, or not? Yeah. Okay, so all right. So now, what does it mean to look through the question? So your tutorial give you a whole compilation of the question. Okay, like for this, right? They will ask, let's say for structure, right? How do they ask the question? Have you been to an interview before?
Yeah.
How do you prepare for an interview?
Uh, I just asked my mom to test me.
Yeah, how does your mom test you?
Mm, I should take the common questions that I usually ask.
Yes. All right. So you will try to predict what are the questions they want to ask you, right? Then you prepare beforehand, isn't it?
Mm,
All right. So that when you go to the actual interview and they ask you, oh, then it's like, okay, this is the one that I spotted. All right. So what you can spot are certain frequently asked questions. That means you know that for this concept, this is how they will ask question. Like
mm,
for this one here. Okay, they want what? So if you look over 10 years, why do we do 10-year series is when you look over 10 years, right? We see or we spot the way that the examiner can ask question for that topic. So when it comes to structure and of the DNA, they will all this. You must know how to recognize DNA structure, all this. All right. Then after that is okay, this is the RNA, this one here. Explain why DNA replication is said to be semi-conservative. Okay, 2 marks, sometimes 3 marks, and then why it occur in a 5 prime, 3 prime direction. Okay, and then the roles of all the different things. So they will, you know, to expect pictures like this. So they will
mm,
draw the replication bubble. Okay, they will ask you to label. So label what? Leading strand, lagging strand. It means that okay, I must look at the picture and I must know which is leading, which is lagging. You see or not? So with that means if they have asked this means you must know what to do. Okay, arrow to show the movement of replication. Wow, that means say I must do all the labeling. I must be able to recognize enzymes. That means the roles of enzyme. Okay, how to actually, why they produce Okazaki fragments. Okay, and then here, how does the replication in the lagging strand takes place? Okay, then this one is what? This one is those picture. And then again, semi-conservative replication. So twice they asked, but they changed the picture. Okay, and the picture, very interesting, you know. 1 year, they use this picture in a horizontal way. Another year, they use this picture in a vertical way. Okay, so they try to, you see, it's the same thing, but they shift and change as if and some students don't recognize it. So that means if you have practiced before, it will make it at least easier for you. All right.
mm,
And then this one here would be the experiment one, the Masterson and style experiment, and then explaining the experiment. Then after that, you see and replication problem. You have seen this picture a few times, all right? They show a few times the role of the telomerase, okay? And then why they need to extend, okay? Yeah, so that means when you want to revise, when you flip through your tutorial and see, oh, okay, that means this is how they will ask question, and then this is how I would then prepare. Okay, I'll go through concept, summarize for you first, and then we're going to do the question so that you can then consolidate. Is that okay? You can go both ways. Either you go at double strand. Oh, there are two strands. Okay, they are bound in a helix, and then how many base per turn and all that. So big structure, you go in. For me, I go small and I go big. Okay, so DNA structure, I will start with the basic unit because this is considered a biomolecule. This part here should be under biomolecule, but in biomolecule, we didn't learn this. We push until DNA, then only we learn this. So actually, this is a biomolecule. So in biomolecule, how did you learn? You learn first glucose, right? Then you learn the glucose joined together with glycosidic bond becomes starch, alpha amylose, and then you have the amylopectin, you have cellulose, and then you have glycogen. Remember? So it's from something small and then you build into something complex. From a monomer to a polymer. So the monomer, the basic unit is a nucleotide. So the nucleotide consists of three parts. This one is called the nitrogenous base. This one here is phosphate. All right. And usually it starts with triphosphate. And then this one is a pentose sugar, or to be specific, we call it deoxyribose sugar. Okay. All right. So now, where is the 5' and where is the 3' end of this molecule? So how do I remember from the nitrogenous base? That's carbon 1'. Okay, the structure. Okay, so when I say start from nitrogenous base, you can actually repeat. I don't mind nitrogenous base, carbon 1'. Just say it out loud, okay? Because it's just the two of us. Because you need that. You need to actually become kinesthetic. Because moving your mouth is also kinesthetic movement. You need movement. Okay, then here 2', then here is 3'. Then here 4', and here 5'. So the top here is actually the oxygen, right? The O, so it shouldn't have the 1'.
Oh.
Okay, because why must you know how to count 1', 2', 3'? Because they will ask you to label. So they will give you drawing, and then say which is the 5' end, which is the 3' end. You must know how to recognize. Okay, so the other one to remember is phosphate. The P is attached to the 5'. So that's another trigger. Okay, 5' end should be next to the phosphate.
Okay.
1' next to the nitrogen base. Okay, 3' is here, 2' is here. So the difference between deoxyribose and oxyribose is at the 2' here is a H. Okay, 3' end has an OH.
Okay.
All right, now bases, how many types of bases are there?
Oh.
What are the bases names?
Adenine, guanine, cytosine, and
Adenine, guanine.
cytosine, thymine.
Okay, so now if I ask you, which is purine, which is pyrimidine?
Oh, purine is adenine and guanine.
Are you sure?
Yeah,
Why are you so sure?
because my school made me memorize like purine is like pure as gold.
Okay, there you see, the moment you put a structure to it, you will never forget. That's what you need to do. Okay, now because this type of thing doesn't make sense, one right? Purine and the Ning Kuan Yin, what's all this? So when things doesn't make sense, why they say pure as gold is so that it makes sense to you. Pure as gold makes sense to you. When it makes sense to you, you remember. That's why there's always tagline, 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 so that the moment you remember the tagline, you associate with something. Okay, so this one is cytosine thymine is
Pyrimidine.
pyrimidine. Okay. So what's the way that you use to remember?
They said pie cut, so there's the uracil at the bottom.
Huh? Pie cut. Oh,
Yeah, so
okay.
it's like PYCUT. They say it's like cutting a pie.
Oh, okay. I remember the Y. For me is my method.
Oh,
Those spaces with Y are pyrimidine.
mm,
So I just remember that one half. Then the
yeah,
other half automatically belong to the other group.
okay.
Okay, now I want you to tell me which one has one ring, which one has two rings.
I think pyrimidine has one ring.
You cannot think. You have to be very sure.
Pyrimidine has one ring,
One ring.
and purine has two.
How do you remember?
I think it's because that time I remembered it as like pyrimidine is the longer word. So there's a
Okay.
smaller one.
Yes. You see, the moment you make the association, you will remember correctly. Now, why is this so important? Because this sort of thing, right, is pure memory work. And you don't want to be in the exam, it's like, oh, is this purine, pyrimidine, purine, pyrimidine, especially MCQ, where they will give you all these things. All right. So things like that, it must be snap of a finger, recall and recall correctly, not agak-agak. No room for agak-agak, this sort of thing. Okay, so this itself will save you time really. Cannot? Okay. Okay, so this is the structure. At least these things must surface when you talk about DNA at the molecular level. Okay, so now the connection, because they will ask you, this one is one nucleotide, right? So now the next thing is they will ask you how the polynucleotide is formed. Now, why is this important? Because in carbohydrate, they ask you, right, the formation of the alpha-1,4 glycosidic bond. They actually ask you how to describe the actual thing. So here, they can also ask you to describe or they can ask you to draw. Just now, there was one question that asked you to draw. And usually, you must know how to draw 4 nucleotide or 2 pairs. So you must know how to draw. Okay, so now you also practice drawing. Now the drawing is like this. You draw this. Even pentagon, you have to practice draw the pentagon. The pentagon don't draw tall.
Mm.
Draw broad. You draw tall, you have no space to write all these things, everything all crammed together. You see or not? Alright? So every time you draw is the same shape. Because what you want is repetition. Every time you repeat the same thing, you reinforce, reinforce. If one time you draw like that, another time you draw like that. So every time you change, is a new information to your system. All right? Such things are skills, exam skills. And then here is the square. Here is the circle. All right? Then after that, I will repeat. So if this is a frequently asked question, It means that when you revise, you will practice drawing until you can do it easily. So how to draw the connection is one nucleotide first and then the second nucleotide. Okay, so this is the effort to practice. All right, so now to connect the two is what? To connect these two is, this is the, oops, this is the 3' end, this is the 5' end. Okay, so you see the nucleotide below come to the 3' end and join here. Like that, join already. Okay, so this is the 5' end and this is the 3' end of the molecule.
Oh, okay.
Okay, now next thing is I need to draw the other strand. And I must know that the other strand is anti-parallel. Okay, this is another characteristic, right? Anti-parallel. So therefore, how do I draw? Okay. Now, some people draw the bases all the same shapes. Is okay. But I draw one square and one longer because I want to show that one is one ring, the other one is two ring. So therefore, I draw like that. Okay, now look how I draw. I draw the bases first. Because why? I have to make sure that this length to this length here is actually constant length. Okay, now then after that, when I draw the pentagon, I have to remember I must point the other way. Okay, then after that, I draw this way. All right. Now, this is provided you can draw upside down. If you can't draw upside down, turn your paper around and draw the other side.
Yeah.
Yeah, that's what you do. Okay, so now, usually when it turned upside down, then the numbering all went chaotic already, right? So always remember that 1' is at the base. So 2', this is the 3' end. This is the 5' end. So therefore, this is the 3' end and the 5' end. So it's between what? The 3' end. Yeah. Okay. And the OH of the phosphate. So it connects like that. Okay. Now, students spend a lot of time drawing the 4 because why? They have no structure in drawing. They try to connect. They'll find that, how to connect the phosphodiester bond. Okay. Now, how do I draw? I draw nucleotide by nucleotide. Do you see that? I draw one nucleotide first. Then I draw the bottom nucleotide. Then only I connect the 3' with the phosphate group. The other side, I do the same. I draw the top nucleotide and then the bottom nucleotide. Then only I connect with that line here. The steps does not change.
Okay.
It's like your CCA, right? When somebody get injured, St. John, how do you test whether the person is still alive or not? Whether you need to do all this? Yeah, there are steps on, right? You learn the steps, right? First is you do this, is it still breathing? Then you're gonna do this, then you're gonna unbutton, then you're gonna count 1,000, 2, 1,000, whatever lah. There are steps, right? You learn the steps, right? Here is the same. Why do you learn the steps? In St. John, all this. Why do you learn? Why must you be taught in steps? First this, second this, third this. Why?
Because you're not able to jump to the end.
No. You know why? In an emergency, when people panic, they don't know what to do. So when we teach you step like that, this, oh, when everybody panic, right? You say, okay, first do this, second do this, third do this. That's why you drill and drill and drill until it becomes natural. When you go and attend to people, you know, oh, first step I do this, second, no, you just automatically, oh, do this, do this, do this, like that. Studying is the same. Okay? Turn everything into proper structure so that you don't go haywire in the exam because the panic will come. So when the panic comes, students will anyhow already. The brain will just go, whoa, whatever that comes, you know. Yeah. So you end up drawing like that. So the student will end up drawing like that, like that, like that, like that. Okay? Yeah. Because panic already. Yeah. Okay, so now draw all the four already, right? Now you have to put in the bonds, the connection. So name the bonds and name the bases first. Looking at this picture, give me the pairs. The top one, here what? Here what?
T and A.
Hmm?
Yeah, T and A.
T and A. So how many hydrogen bonds between them?
I don't know.
Why this one you never go and memorize?
Uh,
You have to know, right? You know that you have to know, right? Correct or not? Two. The next one, what are the bases?
yeah. Um, no. G and C.
Correct. It has to be written in this sequence. So that's why earlier you have to go and memorize which is purine, which is pyrimidine, what base, what base. The number of rings and all that because of this. Okay, this one how many hydrogen bond?
Is it 3?
Yeah, cannot be easy now. After today, 3. All right, don't 3 question mark, 2 question mark. Okay, there's a lack of confidence. Right? 2. A, T, G, C. How do I remember? Okay, if you haven't developed a way to remember, A and T is already one word now. Okay, G and C need one more letter to make it into a word now. Okay, like C-O-G, C-A-G. Okay, yeah. So that's how I remember. That's all. It doesn't have to be very difficult. But the thing is, I never forget.
Okay. Okay.
Okay, all right. You can think of CG as center of gravity. Center of gravity, 3. Up to you, whatever works for you. Okay, now what is the name of this bond? Sugar. If there's a sugar, this would be a
Glycosidic.
glycosidic bond. Okay, then this important one is
Nitrogen.
the bond that connects two nucleotides is what bond?
Oh my god.
Huh? Starts with P.
Phosphodiester.
B. B.
Phosphodiester.
Yes, this is the most important one. Phosphodiester bond. How can you forget? Or how can you not know? That's the thing. Correct or not? It's phosphodiester bond, phosphodiester bond, phosphodiester bond, right? Yeah. So, meaning what? When you just the structure alone, you see, just structure alone, there's so much gap already in you. All right. So, this is, you must know this, you must know how to draw this. You must know how to label all the bases. You must know how to label all the bonds. You must know how to label the 5', 3'.
Okay.
Okay, so the distance between here to here is
20.
too big.
10.
2.
2.
You're very far off. 2. Okay, but the 10 base pair per turn one, this one is correct. Here to here, 10 base pair. But the thing is, the 10 base pair thing is not an important thing. So you actually, I was like, you actually memorize, but you memorize things that are not important. Okay or not? The diameter of the DNA is very important. You must remember the diameter.
Okay.
Okay, 2 nanometer. Anti-parallel. So how do you show anti-parallel? One is 5 prime to 3 prime. The other one is the other way around. So when you draw, all these must reflect in your picture. That means when you study, you're studying all this terminology, right? But when you draw, it's like you have to put into the whole thing. Okay?
The phosphodiester bond from 5 prime to 3 prime.
Yeah. Phosphor means what? Okay, how to remember? If when you forget, right, how do you remember? Phosphor is referring to
Phosphate.
phosphate. So phosphate. Diester means di means
2.
two. Ester is ester bond. No? So one ester bond here, one ester bond here. Lah. Phosphodiester.
Wait, where?
Here.
Okay.
So that's why it's here. Phosphor, one ester bond here, one ester bond here. So that's why I have to draw the box. Phosphodiester bond. So this is one ester. This is another ester bond. So diester.
Okay.
Otherwise, what were you thinking about the phosphodiester bond? What to you is the phosphodiester bond?
To me, it was just a bond.
It's just memorizing, right? It's
I
just
just knew
memorizing.
it existed. I didn't
Yeah.
know what it was for.
Okay. So you see, glycosidic bond, how to remember there is glycosidic bond? Sugar. Okay. Glucose is alpha 1,4 glycosidic bond. So when there is a sugar involved, the bond is a glycosidic bond. So that will help you to remember.
Okay.
All right. General rules. Okay. Then phosphodiester is a must. This is the most important bond in DNA.
Yeah.
So that's why I was shocked. It didn't come out. It came out the other things. Okay. So now, whatever that I put here for you, these are all the keywords you must use in your answer. It must come out in your answer.
Okay.
Okay. All right. Now, the next thing is DNA replication. Any question before I move on?
Um, when you're talking about the phosphodiester bond, you said the 3 prime carbon of a pentyl sugar to the OH group of the phosphate. See how
Okay.
you say
All
it's
right. So there must be an existing 3 prime OH, right? Okay, so here like this, like this, like this. So this is the 3 prime and there's an OH group here, correct or not? All right. So the new one that is coming, this is a new one, right? Is here. The new one that comes in actually has 3. We call this DNTP. Have you seen this word?
Yes.
Yeah. What is this stands for?
Yeah. I don't know, there's a triphosphate.
Yeah. Deoxyribonucleoside triphosphate. So triphosphate means there is PPP, triphosphate. Okay, deoxy means what? Means that this one is a H. The O has been removed. That's why deoxy compared to an RNA, right? So RNA is RNTP, ribonucleoside triphosphate, DNTP. So this is the building block. So when it come near, so this is the 5' end, right? So it's going to move towards this side, move up, move up. So when it comes near, what happens is these two is going to leave. All right. So the P, right? The structure of P is O minus, and then it has OH group, OH group. Correct now? So this OH group of P is going to react with this OH group.
Actually, I don't know where the other O and OH come from.
Huh?
I don't know where the other O and OH come from.
Which one? This two, huh?
Yeah.
HPO3
Oh,
minus,
okay.
H2PO3 minus, phosphate.
Oh, okay.
All right. So that's why the phosphate group has OH, OH, and an O minus. Okay. But the reactive group is usually the hydroxyl, which is the OH. So that's why the OH of the phosphate of this DNTP is going to react. So here it is going to react with this. So when it react, this one, all get out. Okay, and then the H here, OH here, leave. So the P directly bond to the O and the 5' there, like that.
Okay, so the whole OH leaves.
The H leave,
Oh.
like glycosidic like that. The H leaves so that the O is still there.
Oh.
So this one is a carbon here. So phosphate attached to an O attached to the C. So this is an ester bond. And then over here is the 5' OH reacting with the OH. This is another ester bond here. Okay, but this is the new one. This is a new ester bond that will form when it moves up.
Wait, so if they ask you to explain the phosphodiester bond, do you need to name the carbon number?
Yes.
So when I say phosphate,
The 3' OH, all right, so it's between an existing 3' OH group
okay,
with the OH. So, okay, so formation of phosphodiester bond is this. The 3' OH group of existing nucleotide reacts with the phosphate, the OH, the 5' OH of phosphate of the new DNTP. That means the one that is coming. Okay, so this is condensation. Okay, so water is released, and then what you get is, okay, so this is the incoming one, right? So the incoming one is like this and like this. So the new bond is here. Okay, so this is the bond that formed.
Okay.
This is the new ester bond that joins. Okay, so they actually form that one new ester bond. Okay, but usually we say that, we normally say this, two nucleotides are joined by a phosphodiester bond. Okay, meaning what are we saying? This is one nucleotide, this is another nucleotide, so they are joined by the phosphodiester bond. Okay, the P and these two. Okay, now two adjacent nucleotides are joined by a phosphodiester bond. Okay, now what is the term used? Terminology. What is the term used? So we normally draw like that, right? Then we draw like that, right? 5', 3'. 5', 3'. Okay. Now, what's the term used to call this, to refer to this? One side.
Um.
If you want to say this one, right? This thing, right? How do you say it? So you must have the vocabulary, the correct term.
Or is it like template strand?
Huh? Template strand. The other one is not a template strand.
I
No,
don't know.
that's why it depends on when you use it. If you see it as a structure, this one can be called a polynucleotide. Okay, means many nucleotides joined together. So this is a polynucleotide. We use the term strand. One strand. Okay, so when you say one strand, this is one strand. So when you say double stranded, you are referring to the two strand.
Okay.
So this is called a strand. So you don't call it a template when you are referring to the DNA molecule. All right, so the same thing there becomes a template when you talk only about replication. So in replication, the strand becomes a template, but not here. Okay, now,
Yeah.
like you can have many names, right? At home, you are a daughter.
Mm.
You are do you have siblings?
Yeah.
Brother, sister, older, younger?
Sister, older.
There's an older sister. So your role at home is younger sister. You're also daughter. You see it
Mm.
now? So you
Mm.
can have different names to a thing. But I won't call you a interactor at home
Yeah.
because that's not the context. Only
Mm.
school, you're in the interact club. Okay, so there is a role, your CCA, then you adopt that name, but at home you won't adopt this name. So when you're talking about it as a molecule, you won't say that that's a template. It's a strand, a polynucleotide strand.
Okay.
All right? Okay. So because they are twisted, then only you refer to helix. All right? Helix is when they turn. All right? So only when you show that the wavy line like that, then you call it a helix. Cannot? All right? Okay.